I'd like to thank everyone for giving me the opportunity to present and record for you today a version of the lecture that I gave at the Golden Retriever Club of America specialty back in October. I want to point out just the magnitude of the support from the Golden Retriever Foundation. We actually have logged back as far as 1970 a donation from the group, what preceded Golden Retriever Foundation. And since then, we've received $2 million in support, which is just phenomenal. The foundation has supported 75 different studies, which includes the Golden Retriever Lifetime Study. And of course, 1 million of that funding has gone to directly support the Golden Retriever Lifetime Study. So I wanted to give some quick study updates here. Here's enrollment as of October 11th. So I pulled these numbers the day of the presentation that I gave. And at that point, there were 1,355 deceased dogs, sadly. So only 1,188 dogs still enrolled. 130 have been withdrawn, usually not due to a problem with the animal, but typically with the owner. We've lost some owners, and the person who adopted the dog didn't want to continue the study. Occasionally, they needed to withdraw the study because they moved to a different area and they couldn't find a veterinarian willing to sort of take this on. And 371 dogs are inactive. Some of these dogs belong to people who may rejoin the study, but life happens sometimes and you can't get your annual done in time, or something happens. You need to take some time off and then rejoin the study. Of the 1,188 dogs still enrolled, as you can see, most of the dogs are either spayed or neutered. We have a larger contingent of intact male dogs and female intact dogs. And of course, this has changed over time. At the time of baseline, when we fully enrolled the study back in March of 2015, we actually had an interesting even distribution between male intact, male neutered, female intact, and female spayed. Of course, over time, that has changed. And then again, very quickly, this is our geographic enrollment of sort of the distribution. At enrollment, these regions were a little bit more evenly balanced, but it doesn't mean anything that they're different right now, because again, at enrollment, we kind of got lucky and they were fairly evenly distributed in these geographic regions, which are identified by the Centers for Disease Control. Of our primary endpoints, not surprisingly, we have lots of hemangiosarcoma cases. Remember that how we defined an endpoint was a diagnosis of the disease, not a death associated with the disease, as some of these dogs have been successfully treated. Lymphoma, not surprisingly, is our second most highest number of cases of the primary endpoints. I threw histiocytic sarcoma in here because it wasn't considered a primary endpoint at the beginning of the study, but you can see it's actually our third most common cancer. We're keeping an eye on that. It's quite interesting. We didn't anticipate it, but I want to remind everyone we keep track of all of the cancer endpoints in all of our dogs. This little schematic, very quickly, it just talks a little bit about the samples that we collect. Obviously, we collect samples and we give owners the results right away. We also keep some of those samples in a biobank for future use by researchers. So I just want to go through as quickly as possible. This is pretty in-depth. Some of the many studies that are currently active but are sponsored by the Golden Retriever Foundation. And one of these is by Dr. Eleanor Raffin at Cambridge. Many of you may have participated or listened to her Golden Zoomies a few years back. Her whole thing is genetic factors associated with obesity. very quickly, if you see a little yellow asterisk at the end of the title, that means that they are using study samples as part of their project. And Eleanor's still going. She's actually just about ready to publish a paper looking at actually something completely different, which is activity levels and obesity. Another study we have in the works right now by Dr. Michael Povolones and University of Pennsylvania is looking at heartworm and decreasing transmission by mosquitoes through manipulating bacteria, since we know that there's some concern about the heartworm becoming resistant to our current therapeutics. We also have a study with Dr. Josh Stern, who followed up after me at the lecture at the specialty. And what Josh is looking at is if a drug called rapamycin can reverse heart changes associated with subvalvular aortic stenosis in dogs. He is got dogs with SAS and he randomizes them. He gives them SOC, means standard of care therapy, and then they're divided into a placebo group, a low dose rapamycin group, and a higher dose rapamycin group to see if this drug can alter the course of subaortic stenosis in dogs. This next grant is a bit unusual. This grant has been in progress for several years, more than 10 years now, and it is a pathology residency training program that's being conducted at CSU, and this is actually how all of our biopsy samples get adjudicated. These trainees, under the direction of Dr. Kelly Hughes, look at all of our samples along with a supervisor and make sort of the definitive call on the type of cancer present in the samples submitted. So they read all of the biopsy and necropsy samples that come through. So of course they may have been read by somebody else first, but then these guys take a second look. And it's part of our adjudication process to make sure that we're 100% confident when someone comes to us for a sample and it's, hey, we want lymphoma samples. We can feel really good when we tell them that, yep, these are lymphoma samples. This next study, hard for me to describe, but actually super important. Dr. Tamer Ahmed, who's at UC Davis, is actually doing a lot to standardize and make accessible all of our genetic data. I mean, it's a huge, huge database, and it's very, very complicated. And what Tamer's helping us do is kind of organize that. He wrote some instructions for people on how they can access this database, and he's been super helpful in working with us to kind of organize this massive collection of data. Back to something a little bit easier for all of us to understand, which is you guys funded a treatment for ear infections, and particularly biofilms, which are these collections of bacteria that can make it very hard to treat ear infections. And I'm really excited about this study. They're looking at, just kind of assembling their data now. But I want to point out that wide variety of different types of grants that are sponsored by the Golden Retriever Foundation. So it's not always just Golden Retrievers, of course, a lot of them are, but this particular study has ramifications for lots of different dogs. The next study, very important, looking at hemangiosarcoma, and Dr. Feiten is at Utrecht University in the Netherlands, and she's a geneticist, and what she's looking at is the DNA from dogs with cancer and healthy age-matched controls using study samples. And what she's looking for is, are there any kind of genetic markers that could potentially be used to identify dogs maybe early in their life or help make for breeding decisions? Is there anything linked to hemangiosarcoma? Dr. Wood at Guelph, he's been working on another huge project using our study samples, looking at that sort of holy grail, right, of testing, which is an early detection test for cancer. And he's using these little genetic material fragments. They're tiny bits of RNA. If you remember back to Biology 101, there's DNA and there's RNA. And he's looking at little tiny bits of RNA to see, is there something that maybe tumors kind of spew into the bloodstream that's different from normal dogs. This is a really unusual study out of Ross University, again, using samples from the study. And Dr. Hooper is looking at the influence of, she's looking at what are social demographics. So that could be where you live in the world, where you live even in a neighborhood, what kind of house do you live in, what's your income, all these things to see if they have a bearing on canine cancer risk and whether something is modifiable. We don't always have a choice on our salary, but we might have a choice on where we live or how we interact with our dogs, etc. And she's looking at all of that. Another kind of different collaboration is one we have with the Royal Veterinary College, where this group, they have a very large epidemiology group there. And Vet Compass is a sort of a software. I can't describe it any better than it's a medical record, electronic medical record system that kind of binds and is used by many practices in the UK. So they can, if you went to one hospital or a different hospital, they can often pull up your records. And what this group is doing then is it gives them access to this huge amount of data for years, right? It would be, they would have data on routine blood work, when a dog was spayed or neutered, if they had an ear infection, and they're able to look at that. And because of that access, they're looking at data from our study and looking at Vet Compass health databases on golden retrievers, and they're kind of comparing them, like what is different, what is similar, and again, it's at its core a cancer risk study. Another study that's currently, we just funded this one at University of Zurich, and this is really interesting because this group is using a laser to actually cut and collect cancer cells to identify new therapy targets. So actually looking at the cells specifically, which is really unique and really different and hasn't really been done before in dogs with hemangiosarcoma. Another study that's currently going on is we don't think of radiation as really being helpful typically for hemangiosarcoma, but the folks at Animal Medical Center are taking a closer look at single cancer cells compared to normal cells to see if they can find actually a target for radiation. Again, a really unique approach. Again, radiation has kind of been tried, but not very successfully in the past. It'll be interesting to see what they come up with. Dr. Kim, this is one of our more unusual partnerships because this is actually a startup, a biotech startup in Seattle. And these guys are looking at a marker on cells that is called desmoglein 2. And this molecule is a target that's used sometimes in people. There are actual drugs against it. And it's a molecule that helps kind of keep cells together and glue them together. And if dogs have this marker, you may be able to attack this, like they can't grow if they're broken apart. I know that's a kind of weird explanation, but it's an exciting thought and a really different approach to solid tumors. Again, another one looking for early diagnostic tests. This is another one looking at microRNAs in the blood and fusions, which would be also cool, right? If it is in, we know a lot of these tumors bleed. That's often how they're diagnosed. So it would be super if we could figure out a diagnostic test that works in an infusion. Again, more genetic mutations, and these guys also are looking at a mutation that has been known to make cells more sensitive to, if they give this new therapeutic, which is proprietary, so we don't know, with doxorubicin, it can make them more liable to be killed. And what Dr. Kim's doing is looking in hemangiosarcoma to see if they have this particular mutation, because then we could tap into using some drugs they use in people. This is one that I think a lot of the breeders will be interested in. And what Frances is doing is she's using data from our study and developing like a statistical model that you might be able to plug in some information and find out if your, maybe a particular breeding combination would have a higher incidence of hemangiosarcoma and other complex diseases. So it's a super complicated algorithm developing study, but would be very exciting and lots of practical applications right away. Dr. Aaron Dickerson, who's with the group at University of Minnesota, I think we all know that they're a real powerhouse when it comes to hemangiosarcoma research. And what Dr. Dickerson is exploring is, for almost 10 years now, there's been a concept that there are subtypes of hemangiosarcoma. We just can't identify them very easily, but maybe these subtypes actually, you should treat them with different types of chemotherapy. Perhaps they have different prognosis or outcomes. So Aaron's taking a deep dive into that using some tissues that have been banked at the University of Minnesota. Our friend Dr. Jaime Modiano, also at University of Minnesota, is looking at lymphoma. He wants to identify dogs, risk assessment tests, looking at using samples from the study and from other cases of canine lymphoma, and is trying to, again, find a way of identifying these dogs early and then monitoring them and possibly intervening. We have another study going on at NC State, and there's this concept of what's called minimal residual disease. So it's almost like, if you think maybe using an infection metaphor, when you treat any cancer, you hope you get all of it, right? But typically we believe there's maybe a small amount of cells that escape destruction, and then these cells then form the basis, right, of when somebody comes out of remission or an animal comes out of remission with their cancer. And what these guys are trying to do is develop a blood test to detect minimal residual disease after a dog's been treated with lymphoma, because rather than waiting till like a lymph node blows up again, maybe you'd be like, oh wait, hey, we've got minimal residual disease, and so we should do more chemo now. Again, it's a first step. We're still exploring what that means in dogs, but an intriguing idea. Dr. Lecce at University of Milan is trying to find a new way to deliver drugs directly into hemangiosarcoma cells using these little things called extracellular vesicles, which are these little packets. It's hard for me to describe other than they're just like little packets of, you can put stuff in them, and see if she can by delivering them directly to the cells, maybe you could give higher doses, for example, of chemotherapy without the collateral damage we often see to normal cells. Oh, I know you guys have heard about this particular gene. This is the longevity gene, just got a whole bunch of press. And what Rob is doing out at University of California, Davis, is actually looking at our study dogs for this, the ERBB4 variants, and look at whether they correlate with cancer risk and, of course, longevity. Our friend, Dr. Lauren Trepanier, Lauren's done a ton of work on looking at environmental risk factors for different cancers. Lymphoma is especially important to her because she's lost some dogs to this particular cancer. And she wants to know whether, she can pick up different chemicals in the urine, actually. So not just the blood, but urine. And she's looking at urine samples to see if volatile organic compounds and herbicides exposures are associated with lymphoma. So she's working on that right now. Another breeding focused study. This is Dr. Adam Boyko. Adam is the founder of Embark. He's actually a professor at Cornell University, and he's looking again at risks associated with inbreeding. He and a graduate student that was working with him, Dr. Aaron Chu, did this several years ago. They were one of the first people to actually use girls' data. And now they're kind of taking a deeper dive because they're also looking at other, not just inbreeding, but also some other factors like exercise and diet as well. And but they're really focused on still kind of following that inbreeding path. So what do we have upcoming? We have another hemangiosarcoma initiative. We're going to have a second call for proposals and an in-person meeting scheduled for June 2025. We should be launching this in January, collect proposals for a few months. Then, of course, we shut it down. We have people evaluate, and then we will have the advisory board meeting where everybody talks about them and we rank them and choose who gets funded in June. We have, for the first time ever for the study, we've always had open calls where people could access study data and samples, but there was never money associated with it, right? We were like, we'll send them to you, but you got to come up with the money for the study. But for the first time ever, we have money now set aside to fund some studies. So we're very excited. Also, we have new funding for Dr. Rod Page. Many of you remember he was the first PI of the study. And we have a fellowship, a funded fellowship for people studying in oncology, veterinary oncology. That's awesome. And of course, our study team is continuing to work hard on as we come into the final phases of the study. I want to thank everyone for tuning in and listening. I'm so happy to have this opportunity and really grateful to the Golden Retriever Foundation and all of you for all you've done to support the foundation.